Welcome, everyone. Thank you all for joining me, Dr. Elizabeth Janiak, as I moderate this award session, where we will celebrate those that received the Research Excellent Award or the Connors Center BRI Research Recognition Award. We have a total of 10 awardees from various departments and research fields. During today's session, I will introduce each presenter, and then we will play each awardee's pre-recorded presentation, after which we will have a live 2-minute Q&A. So I encourage everyone, if you have any questions for the Q&A portion, please write your Q&A in the chat at any point, and we will help moderate those questions for you. And we are very excited to answer questions live, so please do use the chat to share those questions. This session is being live captioned and recorded. So without further ado, I'm going to begin by introducing our first presenter, Mustafa Ashegan, a postdoctoral fellow, and his presentation is titled Early Detection of Arrhythmia Using Chaos Theory.
Hi, I am Mustafa Ashegan, and this is my presentation on early detection of arrhythmia using chaos theory. Heart arrhythmia is a malfunction in the electrical activity of the heart resulting irregular heartbeats. In this research, we aim to propose an early detection method for arrhythmia based on the theory of chaos. Chaos is a special behavior appearing in some nonlinear dynamic systems, including biological organisms. A property of chaotic systems is that while they are deterministic, they behave like random systems. The only way to distinguish them is applying mathematical tools. Another property of chaotic systems is their extreme sensitivity to the system parameters and history. People usually know this as butterfly effect, that says flying a butterfly in Brazil can make a storm in China. To see a mathematical example, a simulation on a simple chaotic system is performed here. The system is called logistic map and defined here. For some certain values of parameter gamma, this system behaves chaotically. Here we see how the system behaves totally different if the initial value is just slightly changed, confirming the extreme sensitivity property. Chaos can appear in an ordinary system if its parameters undergo a certain change in their values. For example, in logistic map, the system converges to a constant state for gamma equal to 3 and shows chaos for gamma equal to 3.9. Okay, but what is happening in the transition? What happens here is called bifurcation. Bifurcation literally means division in two parts. Here we see the first bifurcation and the second one here. Actually, chaos can be interpreted as a tornado of bifurcations that eventually appears at the end of this transition. Now, with these mathematical backgrounds, we are ready to present the main results. The behavior of arrhythmia seems random, okay, but is it really random? To answer this question, we first wrote a code in MATLAB software to extract peak values of an arrhythmic signal, these red circles here. We then apply a test on the process data and it reveals that the signal is chaotic. Now on one side we have the ordinary healthy heart and on the other side we have chaotic arrhythmia. Okay, so we can hypothesize that the transition from ordinary to chaos, that is actually here from health to arrhythmia, passes through the bifurcation, and detection of this bifurcation can mean early detection of arrhythmia. The validation of this hypothesis that originates from a mathematical basis and has been verified in the first step needs further experiments. Thank you for listening and hope you have enjoyed.
Thank you so much, Dr. Ashegan. As we wait for additional questions to filter in from our attendees today, I have one initial question as more of a clinical research person. If you could help us understand how these results will translate into practical applications for some of our diagnosis and treatment.
Thank you for your attention. Actually, the core idea is that when the behavior of our heart, a normal heart, is exactly a periodic system, and on the other hand, if we can, that we already have done, detect chaos theory in the arrhythmia, the transition from health to arrhythmia is called bifurcation. Actually, the transition from ordinary to chaos in chaotic systems is called bifurcation. And systems with bifurcation behave like ordinary healthy systems, but they are going towards being chaotic. That here means going towards being arrhythmic. The idea for this research is that we can detect this bifurcation. This needs recording the signals, heartbeat rate signals, and then feeding it to a software, and it can detect bifurcations. And this can be early detector of arrhythmia. Actually, it needs verification, of course, but as the first step, what we did as the first step is detecting chaos and not a stochasticity in the arrhythmic signals.
Thank you so much,
You're welcome.
and for clarifying that for us, and congratulations on a fantastic project.
Thank you.
Since we do unfortunately only have 5 minutes per speaker today, it's already time to move on to our next speaker, Akshaya Chandrasekaran.
Hi, I am Mustafa Ashkhan, and
sorry. So we'll be hearing from the pre-recorded presentation of Dr. Chandrasekaran, a postdoctoral research fellow whose work is entitled PAX8-directed nanotherapeutics for high-grade serous ovarian cancer.
Precision therapy in ovarian cancer is challenging as patients usually present with late-stage disease and there are no druggable mutations for targeting. PAX8 is a lineage origin transcription factor that is expressed in 99% high-grade serous ovarian cancers. The proposed project combines nanotechnology, our understanding of ovarian cancer biology, targeted delivery, and CRISPR gene editing to develop an innovative therapeutic strategy for high-grade serous ovarian cancer. This therapy consists of an endogenous CRISPR plasmid that will be PAX8 activated and is delivered to the tumors by packaging it in a biocompatible polymer to form a polyplex. In the presence of PAX8, it binds to the PAX8 binding site in the promoter region of the plasmid and activates it. The plasmid guide RNA is designed to impart a dominant negative allele of PLK1 or ATR, which is synergistically lethal with previously mutated P53, thereby resulting in selective ovarian cancer cell death. In the absence of PAX8, the plasmid is degraded with low toxicity to healthy tissues. Over time, we have optimized several factors affecting polyplex formation and consistently synthesized sub-150 nanometer polyplexes with low polydispersity indices. The unlayered polyplexes bear a positive surface charge and can further be modified using a layer of hyaluronic acid that renders it specific to ovarian cancer through the CD44 receptor and increases half-life in circulation. These are called layered polyplexes. Polyplex transfection is more efficient than commercial transfection agents as observed from the luciferase signal after transfection of unlayered polyplexes containing a luciferase plasmid. The unlayered polyplexes also show increased transfection efficiency when compared to the layered polyplexes, as seen from increased GFP signal in vitro. We were also able to observe time-dependent systemic clearance and accumulation specifically in the tumor-injected regions and calculate circulating half-lives of the polyplexes. In vitro, we observed selective ovarian cancer cell killing in comparison to control cells, and in vivo, after 6 weeks of polyplex IV injections, the mice receiving polyplex treatment lived significantly longer, had visible reduction in tumor burden, decreased HE4 tumor marker, and the desired base edit. In conclusion, well-defined lineage transcription factors have the potential to serve as novel targets for gene editing therapies in ovarian cancer and other malignancies. Combining nanotechnology and gene editing can improve specificity towards ovarian cancer cells, as well as incorporate precision therapy aimed at the underlying genetic architecture of this disease. Thank you.
Thank you so much for that presentation. And I want to remind folks to feel free to write any questions or concerns that you have in the chat. I know that presentation looked blurry to most of us, and this slide at the moment is looking a little bit better. So I'm not sure if we're working on adjusting some settings, and hopefully that will be resolved. So maybe we can just start, Dr. Chandrasekaran, by asking you if if there's any slide you'd like to kind of go back to or to refresh our memory of, since we did have trouble seeing.
Sure, just the previous slide, please.
If we can possibly rewind, I know this was a pre-recorded video, but if we can rewind to her previous slide, that would be fantastic, so that we can all get a closer look. Thank you so much. Go ahead.
Sure. Specificity towards the overarching idea here is that precision therapy, as I mentioned, in ovarian cancer can be challenging because there is not any particular driver mutation that drives this disease. So there is no druggable target for therapy. So what we hypothesize is making use of a common feature among ovarian cancers, which is this expression of PAX8. It's a transcription factor, so ovarian cancers cannot downregulate the expression of PAX8. So we've designed a gene editing therapy that is driven by PAX8 expression of ovarian cancer. And the idea is that this therapy is, or the gene edit is observed only in ovarian tumors because the driver here is PAX8. In other tissues, there is no gene edit that is observed. And just this idea of using transcription factor targeting, where we combine nanotechnology, gene editing, and our understanding of the disease drivers, or the basic disease drivers of any disease, can help in transcription factor targeting and developing more innovative therapeutics for diseases.
Thank you so much for walking us through this visual again. That was really helpful, and I think such an amazing illustration of a truly interdisciplinary approach that is leveraging so many different insights and technologies at once to render a novel therapeutic. So congratulations on this fantastic work.
Thank you.
We are now going to move on to our next presenter, Dr. Yaniko, I apologize, who's a research associate at the hospital and will be speaking on her presentation, Distinct Oncogenic Signatures in Malignant Pecoma and Leiomyosarcoma Identified by Integrative RNA-seq and H3K27AC ChIP-seq Analysis.
Hello everyone, my name is Yaniko and I'm very delighted to present our research work on epigenetic analysis of malignant pecoma and leiomyosarcomas. First, malignant pecoma is a rare sarcoma subtype which is characterized by broad morphologic and genetic heterogeneity affecting multiple organs. Although pecoma is a distinct entity, this tumor shows similarities on histological level with epithelioid smooth mass tumors like leiomyosarcomas, thus making diagnosis challenging. Here we hypothesize that other evented transcriptional level and chromatin state beyond also of TSC1 or TSC2 and EFE3 rearrangement contribute to development of malignant pecoma and differentially distinguish them from other mesenchymal and myogenic tumors. Comprehensive RNA-seq and H3K27 acetylation ChIP-seq analysis for pecoma and 7 other sarcoma subtypes show that pecoma and LMS from the ECGA cohort show most similarities compared to other sarcomas. Further, pairwise differential gene expression and H3K27 acetylation analysis reveal that pecoma have a distinct expression profile and chromatin stains compared to LMS, expressing several melanocytic and lysosomal markers that may serve as novel biomarkers who distinguish these tumor entities. These data were validated in some extent by immunohistochemistry in a subset of sections from both pecoma and LMS. GCAE analysis demonstrated greatest enrichment in the K lysosome pathway in pecoma and in myogenesis smooth mass construction in LMS. Regarding the immune-related pathway, immune infiltration is higher in pecoma versus LMS, specifically for macrophages, confirmed in some extent by immunohistochemistry. In conclusion, our analysis have the potential to facilitate diagnosis and provide a deeper understanding of PEComa and LMS tumor development. Thank you for your attention. I'm happy to reply any questions you may have.
Thank you so much for that presentation. I think we all noticed that once again, some of the poster was blurry. And so our guests, the guests in the audience today were messaging us that they'd love to be able to see it better. And we're working on that right now. So I'm going to ask Dr. Yanniku for you to start where Dr. Chandrasekaran started by just letting us know if there's a specific image that you would love to return to, a less blurry version of, and kind of revisit with our audience here today.
Can you please repeat what is the problem with the blurry?
Many of us were unable to see very well. So if there's a particular region of the poster we could zoom in on to show something that you'd love to highlight for the audience, that might be a good use of your Q&A time.
I think all of them, they are interesting. We can actually probably show the expression data on the right side, up right side there, which actually we see that we have identified from RNA-seq analysis some marker genes which highly express in PEComa, and another set of factors which we have seen that they are highly expressed in LMS. So it's a first comparative analysis between these tumor entities, and also from pathway analysis, we have identified some statistically significant enriched pathways. So if you see down in PEComa, we have seen enrichment in lysosomal, and in LMS we have seen enrichment in smooth mask construction myogenesis.
Thank you so much. And I think just to add one more question before we move on to the next presentation, I'm curious, as you look at these results, was there anything that in particular, what excites you the most when you think about your next steps as you look at these results?
The next step that would be actually interesting to do some functional studies for these key transcription factor and genes that we have identified, but it's a pity that we don't have a cell line for either PEComa or LMS, which is representative of the tumors, and we don't have also an mouse model. But hopefully in the future that we will have both a cell line and mouse model in order to further explore these uniquely genes that we have identified in these tumor entities.
Well, thank you. I think that's a really compelling example of the importance of cell lines and research, and a great example for our audience to learn from. So congratulations on this fantastic work. We are now at time and ready to move on to our next presenter, Abby Hart, who's a clinical research assistant, and will be presenting on dietary restriction in vascular surgery.
All right, my name is Abby Hart. I am a clinical research assistant in the Division of Vascular Surgery, and we've been working on this project for the past few years. So a little bit of background about dietary restriction in vascular surgery. Vascular surgery operations are often hindered by high failure rates and frequent occurrence of perioperative cardiovascular complications. Preoperative dietary restriction is an emerging research field with the potential to address these problems. In preclinical studies, preoperative restriction of proteins and/or calories, PCR, has been shown to limit ischemia-reperfusion damage, slow intimal hyperplasia, and improve metabolic fitness. However, whether these dietary regimens are safe and feasible in the vascular surgery patient population remains unknown. So for this project, we performed an open label randomized controlled trial in patients scheduled for any open vascular procedure. Participants were randomized in a 3:2 ratio to either 4 days of outpatient preoperative protein calorie restriction, 30% calorie restriction, 70% protein restriction, or the control group, their regular ad libitum diet. Blood was drawn at baseline, preoperative, and postoperative day 1 time points, and an extensive flow cytometry panel was performed for all of these time points. During the procedure, subcutaneous and perivascular adipose tissue were sampled and analyzed for adipokine, cytokine, hematology, and chemical blood parameters. Patients were followed for at least 1 year after the date of surgery. Okay. 19 patients were enrolled, of whom 11 completed the study. The PCR diet induced weight loss and BMI decrease without malnutrition. Similar prealbumin levels between time points. Insulin sensitivity was improved after 4 days of PCR. Between diet groups, there were similar rates of reintervention, wound infection, and cardiovascular complications. Leukocyte populations were maintained after 4 days of PCR. So overall, we found that 4 days of the preoperative outpatient PCR diet appears safe and feasible in patients scheduled for open vascular surgery. Although the present study and our previous pilot study are the only studies to date attempted in vascular surgery patients, several other studies have shown feasibility and safety in transplant patients, coronary bypass surgery, and patients for liver resection. So looking towards the future, a larger scale trial should be able to outline whether short-term PCR improves glucose metabolism and whether this can be linked to improved clinical outcomes and wound healing in vascular surgery patients. Thank you to all who worked on this project.
Thank you so much for that presentation. I'm going to kick off with a question. If you could clarify for us as we think about the potential for repetition of this work in other patient populations, how would you describe the vascular surgery population demographically?
Sure. So we are actually currently under IRB review to more or less extend this project, which was ended prematurely due to COVID. So we are looking to essentially just enroll this more or less the same patient population with a little bit more follow-up and a little bit more wound evaluation in that follow-up. But essentially the vascular surgery population, typically those patients also are coming in with hypertension, diabetes, and other comorbidities. So those are also factors that we're looking at that impact how this dietary intervention will help or hurt in terms of wound healing. And the results we see in the blood results as well.
Thank you so much. And so just to kind of further clarify, so this is mostly a more elderly population, and or would you say that it's a mixed age population?
Majority definitely elderly, and that definitely impacts wound healing and recovery post-op. And we have also done projects in tandem looking at frailty and how that impacts outcomes in vascular surgery. So this is kind of looking at possible interventions there and how that could possibly help a typically frail population. And we see dietary intervention as something that is fairly feasible in a population that is older and frail. And, these patients are able to, for example, implement this diet, even if they are diabetic or have other, issues that, might prevent them from a medication therapy or something different preoperative. So definitely an older, more frail population. And we hope that this is something that is possible for that population to implement in the future.
Thank you so much. And congratulations on fantastic work. I'm very excited to see where it leads in other populations as well. So we're now going to move on to our next presenter, Rosangela.
All right, my name is
who is a postdoctoral fellow who will speak about the association of physical activity with bioactive lipids, BMI, and risk of future cardiovascular events, mediation analysis from the vitamin D and omega-3 trial, and Jupiter studies.
Hello, I'm Rosangela, and it's my pleasure to present the work Association of Physical Activity with Bioactive Lipids, BMI, and Risk of Future Cardiovascular Events, Mediation Analysis from Vital and Jupiter studies. This is my disclosure. Our main goal in this study was to examine the association of physical activity with plasma bioactive lipids and incident cardiovascular disease, CVD. We also aimed to evaluate the extent to which BMI, body mass index, might mediate this association. To do so, we analyzed 4 subcohorts from 2 independent studies, Vital and Jupiter, and we employed a 2-stage analysis study design. In the first, we investigated bioactive lipids significantly associated with physical activity. And in the second stage, we carried over the significant bioactive lipids to examine their relationship with CVD in 2 case control sub-studies. The biomarker profiling was performed using a throughput LC-MS. Physical activity was self-reported, and CVD was a composite variable of confirmed myocardial infarction, stroke, coronary revascularization, cardiovascular death, and all-cause death. After finding bioactive lipids associated with physical activity, we performed mediation analysis through BMI, revealing BMI-mediated and no-mediated associations. Then, after finding CVD associations and performing mediation analysis, we detected 15 CVD associations, of which 11 showed BMI mediation and 4 no mediation. In this plot, we can see that bioactive lipids associated with CVD showed a relationship with physical activity, but in the opposite direction. In conclusion, we identified a signature for physical activity-related bioactive lipids also associated with CVD. And although the relationship between bioactive lipids and CVD with BMI-mediated associations were predominant, non-mediated effects were detected as well, implying that more than one pathway may play a role in the physical activity, bioactive lipids, and CVD relationship. Thank you.
Thank you so much for that compelling presentation. It sounds like this is a study that, like any good research study, is leading very clearly to maybe a next study or another set of studies. I'm curious, as you reflect on your findings, if you have any hypotheses about what other pathways might make sense to investigate next.
Thanks, Elizabeth. Actually, we are trying to find other pathways, which is very difficult to find, as these bioactive lipids can be the product of many metabolism pathways. One of our hypotheses is that not the BMI itself is can be the pathway of for some of the BAL, bioactive lipids. It can be the fat tissues it themselves, not the BMI as a composite measure, So yeah, that's our hypothesis for these other pathways that can be involved in this association.
Thank you. So kind of drilling down from that more crude measure of BMI as a measure of folks' mass and size, ratio of folks' mass to size, and looking more specifically at the fat tissues because
one of the molecules that we found that had an annotation is dihomes, which are lipokines, and they are signaling molecules that link the adipose tissue to the organic metabolism. So
based, and they can be found in brown adipose tissues. So maybe there is it's one of our hypotheses.
Well, thank you so much for sharing this fantastic work, and congratulations.
Thank you, Elizabeth.
We're now going to hear from Jack Hu.
Hello, I'm
a research fellow, and he will be speaking about sex differences in the intergenerational link between maternal and neonatal whole blood DNA methylation, an analysis in the Boston birth cohort.
Hi, everyone. I'm Jack from the Brigham and Harvard T.H. Chan School of Public Health. My presentation today is sex differences in the intergenerational link between maternal and neonatal DNA methylation. The intergenerational link in DNA methylation variation could contribute to the inheritance of disease susceptibility across generations. However, no study has investigated the intergenerational correlations in DNA methylation at the genome-wide scale, as well as sex differences. In this study, we aimed to examine mother-newborn correlations in methylation patterns by newborn sex, and to identify genes with significant sex differences in mother-newborn correlations in methylation levels. This study included 396 mother-newborn pairs from the Boston birth cohort. Genome-wide DNA methylation profiling was performed in maternal and neonatal blood samples. We performed 3 sets of analysis. First, we calculated sample-wise correlations between maternal and neonatal methylation patterns. We observed that correlations in methylation levels between mothers and their newborns were significantly stronger than random, suggesting that such correlations were driven by family relatedness. For methylation levels across the whole genome, correlations between mothers and their female newborns were significantly stronger than the correlations between mothers and their male newborns, especially on the X chromosome. Second, we found that among all analyzed DNA methylation sites, over 12% showed significant correlations between mothers and their newborns, however, after adjusting for covariates. 15 of these metabolites also showed significant interactions with newborn sex. They were mapped to genes involved in development, stem cells, signal transduction, and neuropsychological processes. Third, we calculated differences between maternal and neonatal methylation levels. The mother-newborn differences in autosomes were relatively similar between male and female pairs, but were larger in male pairs for X chromosomal methylation sites. When analyzing the associations between mother-newborn differences in methylation levels and the newborn sex, we identified nearly 19,000 methylation sites showing significant associations. Over 3/4 were on the X chromosome. They were mapped to genes involved in neurodegenerative and psychological diseases, neuropsychological processes, development, and sex-specific cancers. In conclusion, mother-newborn correlations in DNA methylation patterns significantly differ by newborn sex, particularly for the X chromosome, with more similarity between mothers and their female newborns. Our findings might offer new insights into the developmental origins of many diseases with remarkable sex differences in risk. Finally, I want to thank my co-authors, the Boston birth cohort, funding agencies. Thank you very much for listening.
Thank you so much for that presentation. And I will ask a question, which is thinking about what the higher correlation between female sex infants and mothers. What are some of the processes that might be at play for males that help to explain this higher association with some more prevalent diseases among males, such as autism and some of the other conditions that you mentioned?
Yes, that's a good question. And actually, in our analysis, we find that some significant methylation sites are mapped to genes related to neurodevelopment, and some of the top methylation sites that are showing sex difference in correlations are mapped to genes that has been related to some of diseases that are more frequently appeared in males, such as intelligence disability in the newborns. And we think that there are very complex regulatory mechanisms from the genotype to the demethylation and to downstream pathways. So I think this is a very complex process. And one of the limitations in our study is that we don't have a paternal demethylation status. So we can only look at the contribution of mothers. So we don't, and in the next, if there are available samples, we could look at the correlation between fathers and male newborns, whether there are similar findings that can contribute to sex differences of this sex differences in the disease origin.
Thank you. So it's a really fascinating area of investigation, it sounds like, for looking into some of the underlying causes and pathways through which we do see such disparate outcomes for infants by sex. Congratulations on this fantastic work, and thank you for your presentation.
Thank you.
Given our limited time, we now have to
Hi, everyone.
go right along.
I'm Jack from the Brigham and Harvard Chance.
To our next presenter. I'm just going to verbally remind participants in the audience, if anyone has questions, to feel free to post them in the Q&A function, and I will read them for you. We really look forward to hearing from you. So we have 4 presenters left. And next we're welcoming Kyrie Kim, a high school student who was a summer student at the Brigham, who will be presenting on mapping the brain-heart connection using brain lesions.
Hello, my name is Kyrie Kim, a student at Thomas Jefferson High School for Science and Technology and a research trainee at the Center of Brain Therapeutics in Brigham and Women's Hospital. Today, I will be presenting my research project on mapping the brain-heart connection using brain lesions. The study of the connection between the brain and the heart reveals the nervous system's capacity to injure the heart. Numerous studies and case reports have shown that nervous system disorders, such as a cerebral stroke, can cause neurogenic damage to the heart. This type of cardiac damage can take the form of cardiac arrhythmia, meaning an irregular heartbeat, or tachycardiomyopathy, a syndrome characterized by inverted T waves that predominantly affect postmenopausal women. While the exact structures involved in the brain-heart connection remains incompletely understood, one common finding is that the damage to the insula is associated with a high risk of cardiac arrhythmia and tachycardiomyopathy, as the region is heavily involved in the regulation of cardiovascular function. However, many case reports show that lesions outside the insula can also be the trigger of both diseases, and the reason behind this is unknown. Therefore, in this study, we use brain lesions causing cardiac disease and test whether they map to a common brain network. We first identified 19 published coordinates of brain regions that, when damaged, cause new onset cardiac arrhythmia after stroke. Since these regions damage different locations of the brain but cause the same set of symptoms, we used the normative human connectome data to compute the functional connections of each coordinate, which would tell us whether the lesions are part of the same brain network or not. By identifying the key hub as a right insula, we confirmed that although all 19 coordinates do not directly include the insula, they are all connected to it. We then defined a brain-heart network based on the connectivity to the right insula. Results showed that this network included brain regions implicated in previous neuroimaging studies, such as the operculum, anterior cingulate, and central amygdala. Lastly, in order to validate the defined network, we calculated the network damage score, which indicated that ischemic strokes causing tachycardiomyopathy damaged this network more than control lesions by p-value of 0.007. In conclusion, our study indicates that the brain lesions causing cardiac arrhythmia and tachycardiomyopathy map to a common brain network functionally connected to the insula. Our findings of establishing a network framework will allow us to gain a deeper understanding of the brain-heart connection and potentially improve the identification of patients at high risk of neurogenic cardiac diseases.
Thank you so much for that fantastic presentation of this exciting work. So I would love if you could orient us a little bit to how this concept of the network contrasts with previous actually, someone else has, I'm going to actually pivot since we have so little time and just read an audience question. I apologize. So we have a question, which is, was it the case that larger lesions caused more network damage, or was lesion location really important?
Yeah, thank you for your question. So when it comes to calculating the damage score of in order to validate the network, we summed the intensity by counting up the t values and then divided by the lesion volume in order to get the damage score. So I think in that case, to my knowledge, the lesion location is what is important, because it would actually physically show if the lesion locations that cause tachycerebral cardiomyopathy is actually physically overlapping with the brain-heart network derived from cardiac arrhythmia. So yeah, hope that answered the question.
So it sounds like the size of the lesion was taken into account in generating that score, kind of as a in the denominator, essentially, yes, of the score. Got it. Thank you. Thank you so much. All right. So to ensure enough time for our last three presenters, we're going to continue. And hear next from Katarzyna Klinowska, a research fellow, who will talk about mosaicism detection rate in tuberculosis sclerosis complex patients without mutation identified in commercial labs.
Hello, my name is Kasia Klonowska. I'm a postdoctoral fellow in Dr. David Kwiatkowski lab in Brigham and Women's Hospital in Boston. I would like to thank the organizers for the opportunity of presenting our research work. Our recent massively parallel sequencing analysis revealed that generalized mosaicism is a common phenomenon in TSC patients who had no mutation identified in a previous genetic testing in commercial labs. In this study, we are searching for TSC1 or TSC2 mutations in TSC patients who had no mutation identified. We have analyzed a total of 144 samples derived from 30 patients with tuberous sclerosis. We have analyzed a median of 5 samples per patient, including different TSC tumors, and matched normal tissues and fluids. In our genetic analysis, we applied hybrid capture massively parallel sequencing for the detection of TSC1 or TSC2 mutations, which was followed by the validation of the identified mutations using a new targeted amplicon MPS strategy, which we have recently developed, with the sensitivity as low as 0.05%. Our study led to the identification of TSC1 or TSC2 variants in 27 out of 30 patients. 24 patients were mosaic and had a median allele frequency in blood of 3%. In this plot, we show the distribution of variant allele frequencies observed in systemic mosaic patients in their tumors and matched normal tissues, which are color-coded as indicated in the legend on the right-hand side. The variant allele frequency of the identified mosaic mutations was enriched in TSC tumors in comparison with normal tissues and fluids. In this study, we have identified a total of 6 novel TSC1 or TSC2 mutations, which were not reported before. These new mutations included large rearrangements like inversion, large duplications, or large deletions. We also observed that the number of TSC-related clinical features correlated positively with the mosaic TSC1 or TSC2 variant allele frequency. In conclusion, despite improvements in molecular diagnostic laboratories for TSC genetic diagnosis, TSC patients without an identified causative TSC1 or TSC2 variant continue to be identified around the world. We have developed an analysis approach for very sensitive TSC1 or TSC2 mutation detection, which is capable of 90% detection rate on such TSC and MI cases. Compared with our previous experience, the current cohort is enriched for large rearrangements. I would like to thank everyone involved in this study, the entire Dr. Kwiatkowski lab and our collaborators. This study was possible thanks to Engels Family Fund and TSC Alliance Postdoctoral Fellowship Award.
Thank you so much for that wonderful presentation and exciting work. I'm sorry, I'm just trying to find my Q&A screen here. My apologies. So in terms of the it sounds like the detection of novel mutations in this population that had not been seen before. What do you anticipate, how common do you anticipate this would be if you were able to replicate this kind of study with a larger cohort?
Thank you for this question. I just wanted to clarify, as I understood it correctly. So you are wondering how common would be novel mutation if we would expand on the number of patients analyzed?
Yes. Would you anticipate like that is there many more mutations that you anticipate finding in the future, or kind of how far down this frontier has this work taken you?
I think that with our current strategy, which enables identification of both small mutations like single nucleotide variants, dinucleotide variants, and indels, as well as large rearrangements. I think that we can anticipate that with expanding our study, we will able to find some more novel mutations and also do very precise analysis with very good sensitivity for a wide range of allele frequencies.
Great, thank you for clarifying that. And congratulations on this work.
Thank you so much.
We'll now move on to Max Liu, who is a doctoral student and a research trainee, speaking about AI-based pathology predicts origins for cancers of unknown primary.
Hi, my name is Max. My project was on using deep learning to predict the primary origins for cancers of unknown primary from histology. CAP broadly categorizes cancers where a primary origin cannot be determined. Most believe identifying the primary tumor and providing site-specific treatment will lead to improved patient outcomes, and this is currently an active area of research. My research hopes to help clinicians in this complex diagnostic process using routine H&E histology so that it can appeal to a wide range of hospitals and services, including in low-resource settings. We developed our model using 22,000 patient size of primary and metastatic size of known primary. We then evaluate our model on a held-out test set of 6,499 cases, as well as an external test set of cases submitted from U.S. international medical centers. Lastly, we identified a multi-center dataset of CP cases, of which 317 cases had a primary differential assigned, and we use these cases for assessing our model performance on CUPs. For classification, the model uses the patient's gender in addition to information from the histology slide and makes predictions for both the primary origin as well as whether the tumor is metastatic or primary. For predicting the primary origin of tumors, the model achieves an overall accuracy of 83.4% on the combined test set of primary and metastatic cases. Additionally, we note that the model achieved a high top 3 accuracy of over 90% and generalizes reasonably well and achieves a similar level of performance on the external test set. Shown on the right, we examined the model's ability to distinguish primary from metastatic tumors across a number of tissue sites. The AUC for this combined prediction task varies by site, but mostly ranged from the mid-80s to 90s. Then we examined more closely the model's performance on metastatic tumors in our test set. The confusion matrix shown on the left shows that the model accurately predicts most tumor origins. Shown on the right, the model achieved an overall accuracy of 82.8% and top 3 accuracy of over 90% on the metastatic cases, which is consistent with the performance on the entire test set. We also built an interactive demo which you can go to and look at different patient cases, the H&E slide, the diagnosis, additional clinical variables associated with the patient, as well as the model's predictions and the corresponding attention heat map. Lastly, on the CEP cases that were assigned a primary differential, we noted an agreement between the model's top prediction and the primary differential in around 60% of cases, while top 3 agreement is over 80%. Based on the language used in pathology reports and electronic medical records, we further divided diagnosis assigned as high certainty and low certainty and witnessed a much higher agreement within the pool of high certainty.
Great. Thank you so much for this presentation and this work and for acknowledging the village of people that it took to bring into fruition. I'm curious, as this seems like a model that is highly accurate by multiple measures. I'm wondering if there were any patterns in what kind of case, in what in like which of those what those misattributions looked like. Was there any underlying pattern in like what kinds of tumors were not correctly categorized or predicted, or what kind of patients they came from, or does it just seem to be random when it does or doesn't work?
Thank you for the question. So I think there's two main trends that we're able to sort of gather. One is, fairly intuitive, which is that for any kind of these, machine learning or statistical models, the more data and sort of the more representative the sample you're going to get, the better accuracy you're going to get, right? So in this case, because the CUP covers such a wide number of disease models and different subtypes of cancers, there's certain rare cases where, even if you dig through the Brigham pathology archive back, decades, you're only going to be able to find, let's say, like less than 50 cases in total. So I think it's for those disease subtypes, rare subtypes, it's not only just about like, whether it gets a high accuracy, but it's also about whether you even have enough cases to sort of properly evaluate it, right? So that's definitely one issue. And, I think that's something over time we can either collaborate with outside institutions and sort of bring that number up, or maybe develop more data efficient methods or more robust evaluation tools to get a better sense of how the model is performing. And I think another thing is just that, this is, using a model that predicts this kind of outcome from just histology, that's just one modality of information, right? So in practice, for this to be useful, a physician would have to look at patient history, medical records, do like multiple IHC panels typically to sort of get a better sense. And because we're doing it only from the morphology, anything that's sort of poorly differentiated, you're going to have a tougher time for the model to correctly get the answer. And sort of just to conclude, right? So the accuracy numbers we report a lot of times are, let's say in the 60% to 80% range, depending on if you're looking at CUP cases or metastatic cases, which is not necessarily that high. But I think what really makes this framework powerful is that it's able to sort of give you, let's say, like, the top 3 or top 5 likely primary origins, in which case you can sort of combine that with a attending pathologist domain expertise to say, okay, out of these 3 or 5, which seem more likely and how we can investigate and probe this further. And I think we also showed that when you consider, let's say, the top 5 or top 3 predictions, the model is actually really, really accurate. So I think that has a really huge potential in assisting physicians to more quickly arrive at the desired outcome, or the correct outcome, and also sort of, using less resources in that process.
Great, thank you so much, and congratulations on this work. So we have just exactly 5 minutes left and one more presenter, so we're perfectly on time. So I am delighted to welcome our final presenter, Michael McCoy, a postdoctoral researcher who will be discussing an miRNA cassette reprograms smooth muscle cells into endothelial cells.
Hello, my name is Dr. Michael McCoy. Today, I will be presenting over some of our most recent and ongoing work in RNA medicine and vascular biology with a project titled A microRNA Cassette Reprograms Smooth Muscle Cells in Endothelial Cells. Non-coding RNAs, in particular microRNAs, have emerged as powerful regulators of cell fate and function. MicroRNAs function primarily through competitive binding of the 3' untranslated region, or the 3' UTR, of messenger RNAs, which results in the repression of gene translation. Spatial temporal regulation of DNA networks by microRNAs regulates cell fate and function. However, the regulatory potential of microRNAs to transform one cell type into another has been largely uninvestigated. Smooth muscle cells and endothelial cells share perivascular interactions which makes crosstalk coupled to function, yet their microRNA expression profiles are quite different. This is of interest as endothelial cells are slow-growing under physiological conditions and have limited regenerative potential, whereas smooth muscle cells are known to be more proliferative. Thus, the transformation of smooth muscle cells into endothelial cells via microRNA reprogramming may be a potential therapeutic application, and is certainly worth investigating. We began these studies with a hypothesis-generating approach and profiled smooth muscle cells and endothelial cells from microRNAs which may be differentially expressed. These microarray datasets generated a list of candidates, and with some screening of high-expression targets, we narrowed down our set to microRNAs 143, 145, 181b, and 146a. Initially, we confirmed by Western blot analysis of cell lysates that endothelial markers were significantly increased in IECs when compared to Cas-Mix. These included VCAM1, E-selectin, von Willebrand factor, and V-cadherin. In contrast, a decrease in the abundance of smooth muscle cell markers myosin heavy chain 11, fomentin, alpha SMA, and calponin was observed. For a deeper dive into the properties of our IECs, samples were sent off for transcriptional profiling. Because much of the transcript was not of specific interest to us, we have to focus on a curated gene set of the endothelial-specific genes. A numeric pathway analysis of top up-related genes in IECs finds an overrepresentation in gene networks associated with angiogenesis and endothelial cell function. In order to investigate whether or not IECs having a therapeutic potential in vivo, we decided to assess their potential in facilitating blood flow recovery in a high-limb ischemia model. Our observations found that IECs improved blood flow in a manner that was similar to HUVECs when compared to PBS sham injections. Furthermore, analysis of the quality of the limb found that the limbs in mice injected with IECs experienced less ulceration and improved healing when compared to HUVEC or PBS controls. In summary, we find the potential of microRNAs as a worthwhile consideration in the transdifferentiation of smooth muscle cells into endothelial cells. Using this cassette of inhibitors and mimics, we transfected smooth muscle cells and they exhibited endothelial cell-like properties following expansion. We found that our generated induced endothelial cells exhibited endothelial-like properties at the transcriptional, protein, and functional level. I would like to give the acknowledgement of this talk to all the members of the Feinberg Lab, but especially to those bolded for their help with this specific set of work.
Thank you so much for this presentation and this really fascinating cutting-edge research. Could you tell us a little more about how the kind of wonderful diagrams at the cellular and subcellular level about what is going on? Could you tell us a little bit more about how on a logistical level, how you performed this, these procedures with the mouse model and what you would see as a next step for that?
Yes, can you hear me? My internet just, I have a stable connection, all of a sudden I do not. But if you can hear me,
we can hear
you. What I would like to say is that we essentially, sorry, can you repeat that question? I got half of it.
I was just curious to hear more about how the mice models worked. We saw a lot of on the subcellular and subcellular level, but for those of us who don't work with mice every day, kind of how does that translate up to the animal level?
Yes, so the essentially we like I mentioned the model, we ligate the femoral artery and look at there's a stage of angiogenesis that we can model, we can monitor within the animal and look at essentially a sort of like a wound healing response. And by just simply injecting pro-angiogenic or anti-angiogenic factors within the bloodstream, you can help facilitate this process. And so by injecting whole cells, we were just curious if they had an angiogenic response that we would expect possibly to be similar to endothelial cells, so that they can aid in not only neoangiogenesis within the muscle, but also then subsequent reperfusion of the muscle following the wound healing response. PBS is just like a saline that we use as our control, so we would expect really nothing to change within that condition. Did I answer your question?
Yes, thank you very much. That was really helpful. And congratulations on this fantastic work. So I just want to congratulate again all of our presenters and award winners here today. Thank you for joining us, and we look forward to seeing you at the next session in just a moment.